cell that is in sort of in this middle space, right? It means it has both E. coli and Bacillus reads. So it likely represents what we call a mixed doublet. And it could result either from physical doublet, where it's sticky and they maybe stick to each other and then travel physically together through our combination of cells. Or maybe it's just a random chance.
Yes, unmuted. All right. So what I'm going to show you now is a bit of a process engineering pipeline, which you typically don't always see in the papers, the successful papers, right? But this is our first historical experiment, just applying split-sig as is to bacteria. Okay, we just wanted to see how the eukaryotic basic method without any kind of adaptations will fare for bacteria. And we've got some data. So here's E. coli, and then we use Lactobacillus rhamnosus for our gram-positive, just for one experiment. And we did have some clusters, but it is pretty poor, right? We only detect maybe up to 100 transcripts, mostly in the 10s for E. coli and Lactobacillus. And also we have a huge problem of losing Lactobacillus. We have about 10% left. We also have lots of doublets. So, and also, I think an even larger problem is all of this reads actually ribosomal. So this is pretty poor. Okay, so that was the first experiment. By the 7th experiment, we have improved. So what we've introduced here is the lysozyme treatment. So now we can break down the cell wall and some detergent permeabilization. We've also attempted to enrich the mRNA, but also I think the major part was played by the specialized RT protocol or reverse transcription protocol. So we're faring better, we're detecting more transcripts, but now we have this huge mess in the middle, right, which is the doublets. So what we figured out is that the cells, after our specialized better for bacteria RT, get really sticky, and so they aggregate after RT, and then they pass through the different wells. And I think they also aggregate even before the RT. So then we did a lot more optimization, and then fast forward to our 11th experiment. So, and this is the actual figure that made it into the microscope technology, right? So we've done, we've left the lysozyme permeabilization, we've switched to mRNA polyethylation enrichment, which is basically putting in poly polymerase to perform an in-cell polyadenylation of transcripts. And it just turns out that it preferentially polyadenylates mRNA over ribosomal RNA, as was published before in an obscure manuscript that we got out. Nobody really knows why, but presumably because the ends of ribosomal RNA are very different. And then crucially, we've introduced the disaggregation and filtration step in the form of mild fermentation, and that helped us reduce all these aggregates in the middle. We also now detect quite a lot of RNA, and we are more balanced between Bacillus and E. coli, getting a roughly equal proportion mix. All right, so how are we doing at the mRNA capture front? Right, remember this plot represented all RNA. So we capture a few hundred mRNA per cell in the median, slightly higher for Bacillus, okay, but also pretty okay for E. coli. So because there were no references at this point, how do we know, is this enough, is this good? Can we see a transcriptional response in cells, right? And then we decided to perform a really dramatic perturbation for cells. We heat shocked the mix, and then we did this trick where we can load a heat shock one sample into one half of the reverse transcription plate, and then the other sample into the other. So now, because we know which well contains which barcode, we can trace back our cells from each sample by saying, for example, okay, this cell originated from this well, and therefore this. Okay. And in this way, we can multiplex by the way many samples in an experiment by just loading them into different wells in a barcode plate. And then we clustered the results, and as expected, Bacillus and E. coli cells clustered very separately. And what's more exciting is that we do see, let's see if it works. Oh, it works. We do see the heat-shocked cells forming a separate cluster, transcriptional cluster from the control cells. And so you will see, okay, let me do a quick intro to the Stiesny plots. You will see these plots a lot. They are basically, H dot here is a cell, and the cells are grouped according to the transcriptome similarity. So when the cells are grouped together like this, okay, here it is, they form a cluster. This means they're transcriptionally similar and distinct from, for example, this cluster of cells. All right, and so basically when we look at these clusters, we can number them and look at the genes that are differentially expressed in all of them. For example, cluster 0 against cluster 1. We do see in both Bacillus and E. coli heat-shock clusters the elevated expression of genes associated with a classical heat-shock signature, like some chaperonins, GRAL-GRAS, CLIP-C protease, DNA K, accessory proteins, and so on. So this is very same for E. coli. So this is very encouraging. We did see this heat shock response. Sorry, in our data. All right, so that was a really dramatic perturbation, though. We basically heated up the cells and we saw the heat shock response. But can we now resolve more complex transcriptional programs with it?
So, question.
Mm-hmm.
So you say you have something like 200 to 300 transcripts per cell.
Yeah.
So how many genes do you have in the E. coli genome that is actually getting transcribed?
We don't know how many transcripts are truly there, or what is the actual, truth.
So but I mean, if you look at bulk sequencing, how many transcripts are getting expressed?
So these are actually the transcripts. So UMIs are unique molecular identifiers. Each one labels a unique transcript and not a gene, right? But we generally detect only a few transcripts per gene. So our gene number per cell is almost the same as our UMI or transcripts number per cell. It's just because our sampling is pretty sparse. And this is the same problem as eukaryotic as RNA-seq, right? It's a 5 to 10% of whole transcriptome, which is, we get this number too, about between 5 and 10% of the whole transcriptome capture. Yes.
Did you ever try to compare this to RNA-fish data from bacteria?
For the heat shock.
Just normal expression or heat shock. I mean, evolving has some pretty good RNA-fish data sets for like a few genes just to.
Yeah, we haven't done the RNA fish. We did a correlation with bulk transcriptome data. So when we just do a pseudo bulk sort of summing of all our genes, and it has a pretty good correlation from a different study. Yeah, but we haven't done RNA fish. Yes.
Maybe I missed something or this thing's in your question, but it looked like you were adding multiple barcodes to each group. Is there a reason for that, or how, and if so, how do you decide how many times you're going to add those on?
You mean how many split pool rounds?
Yes.
Do we do? For now, we don't vary them. We just do 3 rounds with 96 wells because the numbers for us work out. Several, maybe 10, 20,000 cells is typically enough to see pretty good biology, but not prohibitively expensive to sequence and process. But we can definitely scale up. Yeah, we can do more rounds if we wanted to, but we would have to validate this first, of course, that, the barcodes work and so on, but yeah. Okay, so can microsplit resolve complex programs? We've grown, now we're only looking at Bacillus, and we've grown Bacillus subtilis in LB medium 2 times in 2 experiments, and we collected 10 optical density points from OD 0.5 to 6. And a few of them were replicated and a few of them were not, just because of the different people collecting them. And so these are the results of the microscope on 25,000 Bacillus cells. And in this plot, they are colored by the sample identity. And so basically we could capture all of the samples in 2 experiments, and we could have done one, but we just wanted to have replicates. You can probably see that mostly the cells kind of cluster according to the optical density sample identity, and they sort of go clockwise from the 0.5, this blue cluster over there, all the way to OD6. Yeah, let me see if I can, yeah, like this. Okay, and then we can see if we can identify distinct clusters programmatically with this Louvain algorithm. And we can find generally 13 clusters in the data. And most of the clusters, again, sort of overlap with the sample identity, some are not. So one of these examples that is more or less evident right away looking at it is this cluster 9 here. This is a small cluster, and it contains cells coming from this 4 intermediate optical density points. And it contains between 3 to 15% of cells within each of those samples, right? So right there, the cells are doing something transcriptionally distinct, right? But they only represent a small part of this whole population of cells within this respective samples. And I'm going to show you what they're doing in a moment. Right now, I just want to show you also sort of highlight the whole transcription coverage that we have and do regular analysis, and also basically utilize this amazing resource, which is called subtiwiki, on all those regulons. So we, for example, if we take a transcriptional regulator that is in the database, for example, this DEGU regulator here, we can line up all the genes that are known to be activated by this regulator and call it DEGU positive regulon, and then line up all the genes that are negatively regulated, call it DEGU negative. And this is a pretty crude picture, but I think it is somewhat informative, okay? And then we can plot the mean expression of genes in each of these regulons as color of the dot, and then we can represent the proportion of cells expressing these genes in each cluster as the size of the dot. And now we can do it for all of the regulators in the database, and this is the select set of them that belong to this interesting diverse categories on top, and they're shown for each cluster in the data. And so how we can read this plot is 2 ways, maybe. As a suggestion, we can look at a regulator of interest, for example, this carbon catabolite repressor. And we can see that in the early clusters that represent, numbers are more or less representative of the growth stage, with some exceptions, right? So in the early growth stages, clusters 0 through 5, we can see that the CCPN positive regulon, meaning that the carbon catabolite repression is on, and the cells are growing happily and they're eating the preferred carbohydrates. But then around the middle point here in this OD 1.7, the cells switch it off, and then they start to explore alternative carbon sources. And then we can also profile different clusters and compare the activities of these regulators between the clusters. And in this case, these are the 2 different subpopulations of the same optical density sample. 1.7, this sort of intermediate point when the cells are basically at the middle of the growth curve. And then most of those activities are similar, except some exceptions, right here, which are interestingly in the development behavior categories. All right, so switching on from regular analysis to more precise analysis, looking at genes now, and we can immediately notice that most of the clusters are defined by different metabolism changes as the cells explore different food sources as they grow. For example, in the early clusters, they eat the sugars such as glucose, mannitol, and so on, and then they turn on the pathways for different other sugars until this middle point, which is characterized very sharply by the switch from glycolysis to gluconeogenesis, firing up of the TCA cycle. And then towards the later stages, we see all these other pathways turn on. And what is very dramatic here is this cluster 9 right in the middle. Remember, I told you it represents only a small portion of cells from this OD 1.7 sample. And what they are doing is expressing this myo-inositol catabolism genes very uniquely, because none of the cells in the rest of this data set activate them. So the inositol utilization pathway has been more or less characterized to some degree in Bacillus. It contains 3 operons. ILAJ contains the actual catabolic or metabolic genes for the breakdown of inositol. ILR is a repressor of this network, and then ILT is an inositol transporter. And we can see indeed that ILAJ activity is active in cluster 9, really almost nowhere else. ILRS is more or less widespread, and ILT transport is also more or less active in this cluster 9. So in order to validate this, we wanted to know whether it's only, our sparse sampling maybe that creates this illusion of heterogeneity, or is it real? We built now reporters of this inositol utilization pathways, both the transcriptional reporters for these 2 operons and then the protein reporter for ILT. Validated them in glucose versus inositol media. They do turn on in glucose media or inositol media as opposed to glucose. And then basically repeated our experiment with LB growth, and these promoters were pretty silent at the earlier OD. However, at the intermediate OD, we did see activation of them in some portion of cells. So I'm probably going to skip through this or go through this kind of quickly, because we don't really understand why this produces a heterogeneous behavior. But the regulation here is twofold. So there is an ILR repressor that represses both of actually all of those, right? But it's interesting that this ILAJ and ILRS point sort of into opposite directions, and ILR can just land between them, and it's bidirectional actually. But there's also a glucose repression where CCPA, when it's bound to abundant glucose, also represses specifically ILAJ. So you need both of the signal, both the absence of glucose and the presence of inositol to actually activate this full network. And then I think if we were to reconstruct this network and maybe do some dynamic modeling, we could probably figure out why it's heterogeneous, but we actually haven't explored it in the paper. So hopefully maybe in the future. All right, so leaving that intrigue right there, let's switch to from metabolism to the question that I'm actually the most interested in, other rare gene expression states in the data. And right away, we can focus on this little cluster 13, has only 36 cells out of 25,000 or so. So when we look at this gene expression signature in these cells, it is actually extremely unique. Oops, sorry. And most of the genes expressed in the cell, as you can see here, all of these genes starting with an X, belong to the category of PBSX prophage genes. And PBSX prophage is this genetic element that is resident in the Bacillus chromosome. And it's activated when the cell sense DNA damage. The DNA damage induces this 2 operon that make both the PBSX prophage, which is a defective prophage. It doesn't make infectious particles. Instead, it actually chops up and packages Bacillus chromosome in it. And it also can't infect cells, but then the holin-indolazine complex that's also being produced actually lyses the cell and then leaks out and then lyses the neighboring cells as well. So it's an interesting kind of a reaction to DNA damage, sort of a kin killing mechanism. We don't really know what it's for, but and why it's conserved in Bacillus chromosome. But what we can see, what we do know from previous studies, that it is actually activated in a small portion of cells when the cells just grow normally due to intrinsic cellular stresses, and we can actually detect this fraction using microsplit. Okay, so can we I've spent lots of time looking at competence in grad school, so I was really interested in this question. Can we now detect competence with microsplit? And competence cells, to remind you, are these little red cells right here that activate the competence circuit. So we couldn't find this cluster in this general clustering picture because this top level clustering picture shows you the major differences between the different clusters. And these are typically dominated by the larger changes such as metabolism. But we know that competence is a stress response, so we would expect it to be induced at late OD. So we just took these two most stressed samples, OD6 and OD5, and we reclustered them separately. Now, subclustering, that's called subclustering, and it lets us find this more fine structure within the samples. And doing this, we actually found this cluster, 62 cells. And when I looked at the signature, I think that was like the best day of my career because it was a very, very clear signature of competence. Starting with this COMGA, that is the top gene that turned on in this cluster. And all this COM operon is very specific to the competence program, and that encodes the genes that actually make this structure right here that actually pulls in DNA and integrates it, not integrates it, just pulls in DNA. And then there's different machinery that actually integrates it into the chromosome. And we do see it induced. We also see genes that are associated with a specific changes to cell state during competence state that has been pretty well described in the literature, and also some genes that have not been described in the literature. But we also think that they should be associated with competence state. Okay, so that got me pretty excited because we could study this state in its native context, meaning how did the people study this rare gene states is also just overexpressing COMK in the system and then pushing all of the bacterial cells into the state, right, which can produce artifacts. So we could actually detect the system in the cell's native context. All right, so to wrap up this section, microsplit provides unbiased transcriptomic data for 25,000 cells, detects transcriptional responses to changing conditions, and finds rare cell states. All right, so that's the end of the published sort of well-groomed data. And now I'm going to tell you about our ongoing efforts in studying single cell behavior using microsplit. All right, so we validated this method on probably the most well-mixed, condition where we grew the cells in monoculture, and we would not expect actually abundant cellular heterogeneity in such a setting. Perhaps. Naturally, bacteria do not grow like this, right? They grow in a community that can be associated with an environment or maybe with our bodies in the form of the microbiota. And so they experience lots of different factors, such as, for example, very complex environments. So the environment can fluctuate, right? Day from night fluctuations, or maybe the food that we ate. The cells can get stressed or starved very easily. This community also tends to be structured. For example, bacteria can form biofilms that are like bacterial cities. They have architecture and lots of labor division and behavior. There can be many species in this community, right? So there's interspecies interactions that form all kinds of ecology. They can cooperate, compete, and so on. And so all of these factors acting at a microbial community can create a lot of phenotypic states that we pretty much don't have a good idea about in the settings because it's really hard to study. So what we eventually want to do in the long term is to perform the studies of single cell behavior in complex populations such as our microbiota, or perform spatial single cell RNA sequencing of a bacterial biofilm, or perform dual single cell RNA sequencing of the system that is fascinating me for quite some time, where we have a host eukaryotic cell, let's say, and then we have an intracellular pathogen. And both of this have heterogeneity on their own, but then when you combine them during an infection, that creates such a large combinatorial space of different possibilities that it's hard to profile without doing a method that would capture single cell resolution transcriptomes of both, or single cell studies of both. And so we just opened in January, so it hasn't been too much time, but we are starting to collect data in all of these directions. But today, in my remaining time, I'm just going to tell you this little ongoing story, okay? And it's very kind of early stage, and it's not really, we don't have a conclusion yet. And this is the first time I presented. So yes, but I thought it would be fun to let you know about what else can we do with microsplat. So this is this branch where we study a member of human gut microbiota as infected with a phage. And this is the project that we are doing in collaboration with my former UW mentor, Bacillus, and also Neil Day at Ph.D. and his postdoc Norma Morella. So okay. So our gut microbiota, even though bacteria are the most famous members of the gut microbiota, as you can see, they're also the most abundant. There's also all kinds of microorganisms that typically inhabit it. And today, what I'm going to focus on is these two, the viruses and bacteria. And so viruses in the gut typically belong to bacteriophages, okay? They are bacteriophages because they infect bacteria. These are bacterial viruses. And so a little bit about bacteriophage life cycle. They can have two different kinds of cycle. A lytic cycle, where a phage infects a bacteria and then right away forces the cell to produce more phage and then lyses the cell and the cycle continues. Or lysogenic cycle, where the phage can infect the cell and then get integrated into the genome and then lay dormant there for many generations potentially, until an activating event happens and it goes back into lytic cycle. And we're going to focus on the lytic phages today because there's lots of potential as currently investigated for the lytic phages to complement drug therapies, because we do have a really widespread problem of antimicrobial resistance going on. And so in order to stop the cycle where we basically invent new antimicrobials and this bacteria invent new ways to evolve resistance, we can potentially complement these approaches by using combined therapies with antimicrobials and lytic phages. Because phages are actually very host specific most of the time, and they can be used to selectively target the bacteria and weaken them so that they cannot evolve the resistance as easily anymore. And so in this picture, you can see basically the case where bacteria can also develop resistance to phage, unfortunately, which is not very well studied or described at all, but they can do it due to a variety of reasons. But I'm mostly focusing on this picture right here that shows lots of heterogeneity as imagined by the author in the gut, where some cells can carry a phage, some cells are infected with many phages, some cells are not infected, as you can see. And so we don't really know what happens when you just sprinkle phage to bacteria. So we're going today to talk about this bacteria that is a commensal gut microbiota member, Bacteroides fragilis. This is a non-toxigenic strain, 9343. And it's a gram-negative rod. And it can be, normally it's commensal, but sometimes it can be pathogenic. It can be produce abscesses, and it's also associated with early stage colorectal cancer. And basically Norma went to a wastewater treatment plant in Seattle and literally fished for phages in there that can be specific for different gut microbiota members. And so one of the phages that she found was the cyphovirus BF12P1, that's shown right here. That's Norma's picture. That showed remarkable host specificity for Bacteroides fragilis. Even as opposed to a different strain of Bacteroides fragilis, right? So this particular strain. And then she sequenced the phage, and now we have the phage genome, which contains 65 genes with some annotations available. All right, so what happens when we infect Bacteroides with a phage? Basically, this curve shows you different MOIs, so multiplicity of infection curves. What happens generally, as opposed to when the healthy culture, which is this black curve right here, you can see very rapid killing. Within 8 hours, B. fragilis is mostly dead, not really dead because it comes back later. So it does evolve resistance. So we thought this is a really good model to maybe see what are the transcriptome changes in single cells when the bacteria are challenged with phage. So we sampled right now, for now, just to these 2 points with or without phage right before this massive die-off. And we could detect both Bacteroides transcripts and phage transcripts. So this is a little bit messy, but you can see that this is the all mRNA mice per cell that we detect. And TCA is just a bile acid that we also put in just to see how the Bacteroides react to it. And then this plot shows you the phage UMI per cell, and then we can see we detect a lot of them in the sample 3, which was infected in phage, with phage. All right, so time to see some colorful plots. So when we clustered both the infected and non-infected Bacteroides, we can immediately see, there are some subpopulations, but also the phage infected sample forms this very distinct, this orange colored cluster to the left. Okay, but when we looked at what's in there, unsurprisingly, we did see phage transcripts. So basically the big difference is that one has phage transcripts and then the other doesn't, which is probably not that surprising. Okay, we removed phage transcripts from the clustering, reclustered the data. Now we still see separation by phage or no phage. So basically in this way, we can see the single cell response of the host bacteria to a phage infection. And when we look at the genes that are involved, we are now in process of characterizing them, but they're basically showing stress response, some protein degradation, and also the outer capsular polysaccharide remodeling. And I'll touch up on it a little bit later. So now, if we only take the phage infected sample, right? What do you expect when you take some bacteria, sprinkle phage on it, and then they die massively off, right? They all get infected, right? In the same way. Well, likely not, right? So we say, what if we took all of these cells from only the phage infected sample and now clustered them with all genes, both bacterial and phage? And we could find this picture where we see this sort of interesting kind of crescent-shaped structure with 4 different clusters. And the gene expression signature, I'm not going to walk you through this, it's too much. But what is super interesting, I think, is that if we look at this genes from the circular genome of the phage, it only has 65 genes. Okay, it's tiny. And the features that are numbered here from 1 to 65 sequentially, they're actually sort of grouped on this element according to function. So for example, this blue are structural proteins, and they are grouped at this point and at this point, right? And then the DNA repair and synthesis genes are all grouped in the beginning, 1 through maybe 12 elements. And so if we take this sort of circular DNA and then linearize it, right? Showing these elements from number 1 all the way to 65. We do detect all of them, by the way, in the cells. Not in every cell, but in a sample. Now we can see that these phage genes expressed by the different clusters actually group by this location on this DNA, right? And they also correspondingly group by function. So we can see these patterns where, let's say, cluster 3 here, which is on this end of this clustering picture, and at the very bottom shows almost no structural genes, but it shows a lot of the DNA repair and synthesis genes. And then we can also just pull all of the genes with annotated function from the phage genome and replot this picture. And now we can maybe start putting labels and meaning to those clusters. For example, cluster 3 likely doesn't have structural proteins, but it has replication. genes. It does have integration genes and the nuclease and chromosome packaging somewhat, only one of them. So we hypothesize maybe that is an early cluster or an early stage of infection. The cells that are not making phage capsids yet. They are just, maybe the phage just integrated in them and is just replicating. But then, and so in the same way, we can look at all of the other clusters and sort of draw this trajectory where we have early infected cells in different cycles of phage infection, right? In different stages of cell infections, of phage infection cycle, from early to intermediate and to late. All right, so that's what we've done so far, but then of course we're also interested in this trend where we see the change in CPS, or capsular polysaccharide genes. So capsular polysaccharides are a very interesting group of genes on top of Bacteriales, where it is actually known to undergo phase variation, meaning that each of the Bacteroides cell can exist in 8 different variants of CPS. From A to H through this action of a recombinase, MPI, that actually goes in every single cell and sort of stochastically flips the switch at the promoter, and then whichever type of CPS operon gets activated, it then represses the rest. So it is thought that these operons are mutually exclusive, and so the cells can exist in all these different variations. However, in most of the data, there is a regulation of the MPI so that MPI can preferentially switch on one of these types corresponding to different conditions. For example, Bacteroides fragilis in the gut will have different capsular polysaccharides than Bacteroides in the lab or in the mouse. And so in our case, we do see PSH prevalence and slightly less PSH prevalence in slightly fewer PSH cells in the phage sample. And this is just the single cell plots showing you the PSH and PSB are the predominant capsular polysaccharide types that we find in Bacteroides. And then, of course, we are literally right now performing the temporal profile where we want to capture this emergence of phage resistance and see what are the single cell transcriptomics changes in the cells. All right, so with that, I'm done. I'd like to acknowledge the people in the lab, Carl Geiser and Sophie Sploss, for doing all of the microscope experiments. And also my former mentor, Georg Stilich and Ruben Polo, and Neil Day from Phenotypic, who is our collaborator on this Bacteroides phage project. And also Leandra Bretner, Luana Paleologu, Timothy Yu, and Norma Morella, who helped with different aspects of collecting this data. And this is our affiliations and the funding from DOE. Okay, with that, thank you.
So one, you're doing the sorting of the different clusters of the phage expression. How do you account for some cells that have different numbers of phages that would be attached to it, with eventually some sort of solution, and how you separate each cell in different groups?
Yeah, so this is a really good question. We don't have yet a way to segregate them into groups with, MOI, right? Some have maybe several pages. Well, and it's really hard to, we capture them at a specific point, and then we fix them, and then the pages multiply, right? So even if only one page infected, then, it would have been multiplied. So you actually don't know whether it's finished or MOI or the stage of replication. Yeah, so I think that's a fair comment. So maybe some of these clusters may represent just the phages that are just gotten in there, as opposed to the phages that are in the latest stage of the cycle.
The other comment, so in part of your protocol, you mentioned the segregation group. So does the segregation ever remove to make the phage except separate in the cell if it's in early stage infection? Like you have partial subtilis cells in your infected condition that don't have big genes, but would be something in sort of infected area?
Yeah, maybe we could somehow fluorescently label the page. I don't really know how we can engineer this page. Then you could put sort of sort the cell if you had expression. Yeah, but I don't know if somebody else has an idea how to segregate this page infected versus non-infected. I mean, there must be some correlation to them.
So when you do the single cell sequencing, you basically pull out these different clusters that have unique gene expression signatures.
Mhm.
You then also know how many cells fall into each cluster. Right?
Yes.
So if you now make a fluorescent reporter strain that have one of these promoters or maybe multiple promoters from a specific cluster expressed, and then you look at a microscope, you recover the fraction of cells that.
No, so we were really puzzled by this first initially, because we see much higher proportion of cells expressing, let's say, pronositol, right, and we did this experiment. Much higher proportion express the fluorescent proteins than we saw the transcripts. But remember, the fluorescent proteins are extremely stable. So and we also saw a shifted temporal profile. We saw the emergence of this population later, and it persisted way later than we detected transcripts for it. So I suspect that this is just that like the YFP, CFP were very slowly diluted out, where the cells are not producing transcripts anymore. And that's why it's sort of artificially also maybe inflated this proportion that we are seeing with fluorescence.
If you would do RNA fish on these cells, would you expect to then capture?
Yes, I think the RNA fish should be probably a better method. We haven't done it because we just, I don't know how to do it, but I think if we did, it was probably a better experiment. I know how to do fluorescence microscopy, and that's basically what we did as a validation. Just basic idea, right? That it's heterogeneous or not. Yes.
When you look at these smaller clusters, like the provenance cluster with like 67 cells in it or whatever, is that enough to capture like a quantitative look at all of the genes in the genome with that few cells? Or I guess how many cells do you need to, I guess, have a more complete view, like most of the genes at least are?
Yeah, it's an excellent question, and it's a very hard question. And I think in general, this sort of clustering techniques, they're not quantitative, they're qualitative groupings, let's say, and then, they're suggestive that these genes may be differentially expressed and they are grouped together, but they will not, so the resolution, the depth to which you can profile the genes activated will depend on the actual sensitivity of your method and the number of cells. And for us, so confidence is known to be inducing about 165 genes by the confidence description master regulator complete. We are detecting maybe 25 with this numbers, right? The 67 and so on. So if we were to increase our resolution and maybe profile the major part is like profile more cells from these later genes, because later these did not constitute a major part of that. Also, the other part of the issue here is that our resolution drops as the cells enter stationary. So basically, we detect more transcripts when the cells are in their happiest state, when they're just beginning log growth. This is when we have this like benchmarking experiment. But then as the cells actually exhaust nutrients and they go above certain OD, then it just drops. So we go from several hundred UMIs to maybe like 30 for this later. But actually, this kind of highlights the power of this single cell RNA-seq analysis tools that many people have developed over the years, that they were able to actually capture this confidence program at all from this very low kind of resolution profile, right, on maybe like a couple thousand cells in total from this varieties. Yeah, so that's really a tribute to this to this. Yeah.
So going on the rare cell, there is this might be a bit, there is this idea of persistent cell, like it was popularized for the Nathan. And that idea is also critical into the cancer. I now talk about Bacterial and persistence and things like that. So I find this, I mean, you can explain better than I can, but these are cells that actually don't grow in exponentially growing culture, they actually set aside and like a backpaging kind of strategy. Like, did you detect these cells? Is there a program there to assist her?
Yeah, excellent question. Yes, one of the things that comes to mind how we should, leverage this technology is look at persistent cells. As I learned, it is a very big field, and it's different for gram-positive and gram-negative organisms, obviously, or not obviously to me, but obviously to people. And so we tried. I think the larger question for the technology is, like you said, is there transcriptional order? Because if the cells are just dormant and they don't do anything, we will not see anything. Because we need some transcripts to be there for the cells to, form a cluster for us, right? If we don't see transcripts, we may not have just permeabilized the cells, or maybe it's, the cell has, disintegrated through the protocol. Not all of the cells make it. Also, we filter we filter a lot of cells out as we do. So we do not have this capability to detect a state based on negative, right? Not having something. We need something to do that. But having said that, not all persisters actually are dormant. Turns out in the salmonella system that I'm studying as my system for studying this eukaryotic host cell infected with intracellular bacteria. This bacteria, salmonella, form persister cells with the eukaryotic cells, macrophages to be complete, and these have been shown to have metabolic activity. They have been dividing, but they just tolerate antibiotic for whatever reason. And the exact reason is not really known, but people have shown that they are not just dormant. Because they cannot be dormant within the eukaryotic cells also. They need to actively be more in the environment so that they're not just killed by macrophages. And so I think this is a pretty good system for us to be looking into persistent cells, and we are on it. We tried a different system, and Bacillus exposed to antibiotics that target the cell wall, and we weren't able to figure out a cluster that would have a clear signature of something that would correspond to that state. Yes, but that's ongoing. Yeah,
So I had a question to follow up on that same kind of rare cell state idea. If I remember correctly, one of those states was represented in multiple amount of the OD progression.
Yeah.
It was a very small subpopulation, but it was found in several successive OD positions.
Yes, which is a nozzle program.
It was specifically okay. I thought there was a couple of them that did that. Was it?
Yeah, well, maybe it's just not presenting, but that was like an island in the middle of that. And that is a nozzle.
So is that expected essentially through that particular range of ODs? So what is it about the density in that region, that specific region, that makes it persistent and separates it away from all the other cells? So.
This is an excellent question too, and like we said in the paper, a lot of the metabolic changes seem to follow the same kind of in the same vein, right? They follow the almost a temporal order. So we see this particular preferred sugars light up first, and then this one, and then this one, and this one. And then inositol is also within this very narrow ranges. So what I think is happening, so inositol, here's the conundrum. Inositol is not supposed to be known, right? People have used before when they characterize the inositol network, they've used LD as a negative control in their sense. So they haven't picked up those genes yet, generally. So what we think is that there's several possibilities. There may be truly inositol in the media through the yeast extract, but in a very, very trace amount, right? The yeast makes inositol as a precursor, hydrogen inositol, and so you may have some. And then as the cells go through this progression of metabolic changes, they deplete their preferred sugars by the OD about maybe 2-ish. And then they turn on, when we do see this in the transcriptome. Yeah, I can even like go there. We do see this really rapid switch, a dramatic switch where I said they switch from glycolysis to glyconeogenesis. That is actually a very big transition point for the cells. Sorry about that. Yeah. So here, basically this is a really big point where you see this, for example, what I call flux switch, where there's two, the A and the B that switch to flux. Basically from the top carbohydrates into the TCA versus from TCA up top. And so there's also stress response genes turning on at the time that corresponds to this particular state right here. So it's like basically when this state suddenly go to here. So it's a really big transition. And at the same time, you see these other states, and this is inositol. And so what we think is that this is where your carbon catabolite regression turns off, and the cells suddenly start sampling stuff. So maybe some of the cells, so one possibility is that the cells sample, some of the cells sense in trace amounts of inositol, and that is enough to activate their network, maybe kind of noisily. And then they eat them up, and so that's why it shuts down after some time, so that it's only active in a few time points. So that's one possibility. The other is that they don't actually sense inositol, but something else that resembles the inositol derivative, which is actually the molecule, the signaling molecule that turns off ILR in this network. This is this, right? So here I'm showing ILR binds inositol, and that releases the regression. Really, it's not inositol, it's a derivative that is down the catabolic pathway, and it actually kind of looks like another sugar. I forgot which one, probably monos. So maybe some cells do get erroneous inactivation of ILR by another sugar. So that's another possibility. The third possibility that there's no inositol in the media. We cannot know without measuring, but if there's no inositol in the media, that would actually be super interesting to me, because it would probably indicate anticipatory behavior in the system. So Bacillus subtilis is a soil bacterium. It's evolved to live in the soil and go through the cycles of feast and famine, so to speak. And inositol is an abundant resource in soil, and Bacillus is one of the few microbes that can actually process it and subsist on inositol as a sole carbon source. So we think that the Bacillus may have evolved to anticipate, in hard times, maybe devote a fraction of cells to turn on this network and see if they can actually, survive better if there's inositol around, not, wasting time on sensing and activating this network. So, and there's theories on this anticipatory gene expression regulation in many books, but it's a little bit, we don't have proof. So yeah.
Two more technical questions. So when you do the cross-linking to the formaldehyde,
Yeah.
does this impact the transcripts you can detect?
So that's a good question too. I'm sure it is. I'm sure cross-linking doesn't do it already. We obviously cannot do our method without cross-linking or without fixing this. So we tried a bunch of fixes that did not cross-link. The cells are structurally not very sound and so we lose a lot of cells. If we are not cross-linking, so for example, if we for eukaryotic cells, they can actually be fixed with methanol and you'll get the result. But formaldehyde was slightly better. If I remember correctly. For bacterial musters, for now, yes, formaldehyde. But we are exploring. Okay, so the method is not